Wouldn't it be great if we could make cancer therapies that were both more effective at treating tumors and safer for patients? That's just what myself and other researchers are trying to do by finding proteins that are really unique to tumor cells, proteins that are kind of like red flags for cancer. With the right red flag, we can make therapies that make previously incurable tumors treatable. Unfortunately, finding the right red flag is really easier said than done. You see, the tumor cells were once the patient's own healthy cells. And because of that, it's really hard to specifically treat and drug a tumor without also affecting the patient's healthy cells. That's why traditional chemotherapies are so hard on patients. Those therapies are kind of like using dynamite to take down the wall of a house. Yeah, it's going to work, but there's going to be collateral damage. You're going to end up taking out other walls, ruining some furniture. It's just a mess. What we really need if we want to take out a single wall are power tools. And in the past decade or so, researchers have started sourcing power tools and inventing them based on proteins made by your own immune system. Antibodies are just those power tools. They're a class of proteins made by your immune system that bind other proteins, typically these kinds of red flags we're talking about, quite tightly. Antibodies are made to recognize red flags on everything from viruses to bacteria to ideally tumor cells, though obviously that's not always the case, or your immune system would just clear the tumor. Sometimes your immune system needs a little help. It needs the plans for the remodel we want to do. And we can do that as researchers and scientists through either clever tricks of immunization or using what we call synthetic antibodies. And once we have an antibody that recognizes the red flag that we're interested in, we can engineer that antibody therapeutically. We can have it deliver a cytotoxin, a drug, a toxin that's too potent to administer on its own to a patient. We can use the antibody to deliver it directly to tumors. Or we can engineer antibodies to recruit immune cells to the tumor and try and clear the tumor that way. These antibody-based therapies have been really successful in the clinic, but are still limited by finding those right red flags. Because the tumor was once part of the patient's healthy cells, it's really difficult to distinguish the tumor from the patient's own cells. And even when you find a red flag that is unique to the tumor and not found elsewhere in the body, it can be hard to find a red flag that's common across a wide variety of tumors, given that the tumors are as diverse, if not even more so, than the patients from which they originate. And so a lot of the red flags that we've identified so far result from the genetic mutations that occur inside of a tumor. However, these can be very specific and individualized and not always applicable to wide populations of patients. However, they have had success in the clinic. But what I'm interested in are finding red flags that are more broadly applicable, red flags that don't originate necessarily from the mutations in the tumor cell, but from the really weird metabolic environment that tumors create. Tumor cells' really only goal is to grow, and they'll do that at any cost. And so all the tumor cells inside of a tumor are all competing with each other for the same resources and with the patient's healthy cells to grow and expand. And they do this to the point where they literally start to eat themselves in a process called autophagy, literally to consume oneself. And as a result of this autophagic process, tumor cells undergo molecular level remodeling of their surface and of the environment around them. And I believe that some of those remodeled molecules are just the red flags that we're looking for. And so in my research, we're first trying to identify those red flags that result as a process of the weird metabolism of tumors. Then we endeavor to confirm that those red flags really are tumor specific and aren't found anywhere else in the body. Third, once we confirm that our red flag really is just a red flag for the tumor, we engineer antibodies that recognize these red flags and then make them therapies. And I'm optimistic that using this approach, we're going to be able to identify red flags and develop therapies that are able to treat wide varieties of patients and make previously incurable cancers curable. Thank you.